All right.
Oh, the cartoon.
So we
That's a great name.
we're going to start by talking about sort of the processes that organisms use to make gametes. And then we'll start talking about reproduction of various types of organisms, eventually working our way to genetics and genetic engineering and things like that. So today we're just going to focus on the process of meiosis. So what have we already learned about related to this? We learned about mitosis. So in mitosis is one form of cell division. It's the form of cell division that makes all of our new body cells for us to grow or to repair tissue, to replace cells. Meiosis is sort of a specialized type of cell division. It's only really used for one purpose, and that purpose is to produce the gametes. That's not what I want to do. Gametes are special, like we said in our question today, gametes are specialized cells. And they're specialized because they have half the number of chromosomes. So the purpose of meiosis is to take normal cells, which we would call diploid body cells, and from them produce a special type of cell called a haploid gamete. Diploid means that the chromosomes are found in pairs. So in humans, we would say the diploid number is 46. Meiosis takes diploid cells and produces haploid cells that have 23 chromosomes, just one from each pair. So we're going to talk about the steps in the process and how it works, like we did for mitosis. So this process is called gametogenesis. That's the process of making gametes is called gametogenesis. And it happens in all sexually reproducing organisms, from plants to animals, fungi, and so forth. It's similar in some ways to mitosis, but also different in other ways, and we'll talk about some of those differences. The name of the phases is the same. It goes through prophase, metaphase, anaphase, telophase. However, it does each of them twice. So the cell divides twice in meiosis. So we'll talk about that. They're named with Roman numerals usually. So the first division is called meiosis 1. The second division is called meiosis 2. And the purpose of making these haploid cells is so that when they recombine in fertilization, they bring the chromosome number back to the typical diploid number. So just like in mitosis, an interphase is kind of the in-between phases. When the cell's growing, when the cell copies its DNA, that happens during this interphase. Remember, before cell division, the DNA makes a copy of itself. Bless you. What's the next phase that comes after interphase? Prophase. All right, so in prophase 1, the first prophase, things are a little different. So we need to talk about this diagram first. So the chromosomes that we have in our body cells 46 chromosomes. Half of them came from our mother and half from our father. Half came from the egg cell, half from the sperm cell. Okay. And so they come in these pairs that we call homologous chromosomes. So for example, in this diagram, this might be the chromosome that came from the mother, and this one is the chromosome that came from the father. They're not...
Attention all staff and students. At this time, the hold in place is discontinued and we are back to our normal schedule. I'd like to thank everybody for, again, taking these drills seriously. We at this time are back into our normal schedule. Thank you.
So these two chromosomes, this one we said come from the father, this from the mother, they're not the same. So they have the same kind of information. For example, maybe on this part of the chromosome there's a gene for eye color. and then here on these chromosomes is a gene for hair color. It would be the same type of information on each chromosome, but because they came from different parents, it might have different information. This might be a gene for blue eyes, while this is the gene, the DNA that codes for brown eyes. And this maybe is for red hair, and this is for blonde hair. So they have the same kind of information, but not necessarily identical. So those are called homologous chromosomes. The two halves of the X, they are identical. They're called sister chromatids. So they're exact, when we say the DNA makes a copy of itself, this half just is copied to this half, and they're held together by that central. So this and this are identical, this and this are similar. All right, so in meiosis, the way the chromosomes line up is different. Because what happens in meiosis is they sort of match up with their homologous partner, and they line up next to each other. That is called synapsis. And when the chromosomes align with their partner, it forms what's called a tetrad, because there's sort of 4 strands. Tetra meaning 4. So there's sort of 4 strands here. And when they're aligned like this, something called crossing over can happen. Because the chromosomes are very close to each other, and because they're homologous and very similar, they can actually exchange pieces. And a piece from one of the homologous chromosomes can break off and be transferred to the other and swap with it. Like you see in these red and blue chromosomes here, they've exchanged pieces down here. And this is something that adds variation. We say that in sexual reproduction, more variation is introduced. And this is one of the reasons. Because the chromosomes, sort of like shuffling the deck of cards, that you're mixing them up, you're swapping pieces, so that these chromosomes are now different from each other. You have new combinations of traits. So that happens in meiosis. It doesn't happen in mitosis. Because in mitosis, the chromosomes don't form these tetrads. So they gather together with their partner. They line up on the equator of the cell during metaphase, the middle of the cell. Spindle fibers attach to them. And then those spindle fibers begin to shorten, pulling the partners apart from each other, separating the homologous chromosomes so that one from each pair goes to each end of the cell. That happens during anaphase. So in anaphase, these homologous chromosomes will get pulled apart from each other. Once they've been pulled apart and they've reached opposite ends of the cell, a new nuclear membrane will form around each set. And that's the end of the first division of meiosis. If we look at sort of a diagram showing all these steps, we see this is meiosis 1. This would be during interphase. Chromosomes copy themselves. In prophase, they match up with their homologous partner in pairs. They might cross over, like these two have. In metaphase, they line up together in partners along the middle of the cell. In anaphase, they start to get split up. So these chromosomes will be one cell and these chromosomes in the other. So the homologous partners have been split in the first division. The second division is basically identical to mitosis. So in prophase, okay, the daughter cells actually have half the number of chromosomes. If we're using this diagram to look at, this is the original cell. It had 4 chromosomes in it. They copy themselves here. They got together with their partner. They lined up in the middle of the cell, and then they got separated. these two went to this cell, these two went to this cell. So now, there's only 2 chromosomes in this cell, half the number that were here. Okay, so those daughter cells are going to go through the process and split again. They line up in the middle. The 2 halves of the chromosome get pulled apart by spindle fibers, moves to opposite ends of the cell, and then finally a new nucleus forms around both of them, resulting in 4 cells being produced. Meiosis takes 1 cell and actually produces 4 cells from it because it divides twice. So if we look at an overall diagram showing it, okay, this is a continuation from the 2 slides ago. Chromosomes that were separated line up, go in the middle of the cell. The 2 halves of those chromosomes split apart. They get pulled to opposite ends of the cell. The cell eventually splits, and what you end up with are 4 cells. But each cell only has half the number of chromosomes as the original. If we think about meiosis in animals, it's a little bit different in production of sperm cells versus egg cells. So on the left of this is what we call spermatogenesis, producing sperm cells. So we start the same. We have 1 cell, a diploid cell. We abbreviate that 2N. It's got chromosomes still found in pairs. It goes through the process of meiosis 1, okay, produces 2 cells. They then split and each form 2 more cells, so we end up with 4 cells. Those 4 cells actually are each different from each other, and each of them goes on to become a sperm cell. However, in egg cell production, it's called oogenesis, the original cell splits once, but it splits unequally. One of the cells that's produced ends up with most of the cytoplasm, and the other is a tiny little cell. And then they each split again, and again, there's unequal splitting of the cell. So what happens is you have 1 large cell that's a viable egg that could be fertilized. These other 3 things produced are called polar bodies. They actually just sort of break down. They could not be fertilized. So when egg cells are being produced, 1 cell divides through meiosis, but actually only produces 1 viable egg cell. In sperm production, 1 cell goes through meiosis and produces 4 viable sperm cells. Sperm cell has the chromosomes in the head of the sperm, where the nucleus is. It then has a little sort of molecular motor with lots of mitochondria, and attached to that is the flagellum.
Why is that flagellum so long?
Because it needs to make its way all the way from where it's deposited
It's like
to where it's going to fertilize the egg, and that's quite a journey for a tiny little sperm cell. All right, so let's just, we're going to watch this video real quick. It'll just show you in an animated way the process of meiosis. It helps sort of, I think, to visualize it a little better when you, and meiosis produces 4 daughter cells. Now in animals, 4 actually become sperm cells and only 1 actually becomes the egg cell. So those are the sort of a comparison between the 2. Any questions about that? All right.